Right, thank you everyone for waiting. Let's get started with our webinar. Good afternoon, everyone, and thank you for joining us for today's webinar. I'm Cassie, Marketing Supervisor at GenScript, and I'll be the moderator for the webinar today. So a little background about GenScript for those who are new to us. GenScript is the leading life science research and application service and products provider in various fields, from basic life science research to translational biomedical development, industrial synthetic products, and cell therapeutic solutions. In today's webinar, we have Dr. Yu Yifan, our project manager of cell therapy products, to present on how to generate highly purified, functional, and expandable human T cells or NK cells from PBNC or apheresis with CytoSinct reagent and platform. A little background about Dr. Yu. Dr. Yu received his PhD degree in the Department of Chemical Engineering at the University of Florida, United States. During his PhD, his research work was focused on the modification of contact lenses and using it as a novel drug delivery platform for the eye diseases. At his last stage of PhD, he joined Dr. Tenmay's lab, focusing on the quantitatively analysis of the cell nuclear migration and stalling forces and its impact for the cancer treatment. So after his PhD, he joined GenScript as a senior scientist and currently serves as the project manager for the cell therapy products portfolio. He is well experienced in the cell therapy processes, especially to the applications of the key raw materials, including cell isolation reagents, cell activation reagents, and cell isolation instruments. This webinar will take around 40 minutes, and we'll have about 50 minutes for the Q&A session after the webinar. You can type in your questions in the chat box and we will answer them after the presentation. If we didn't get to answer your questions during the Q&A session, we will email the answers back to you after this webinar. So now, without further ado, we will let Dr. Yu to start his presentation now. Dr. Yu, over to you now.
Sure, yeah. Thank you for Cassie's warm introduction. So, hi everyone. My name is Yifan Liu, the project manager of the cell therapy products from GenScript. So today I will briefly introduce the current situation of the key raw material of the cell therapy, and also share the information of newly launched cell isolation platform, our CytoSinct technology. So I think everyone here probably seen this picture for more than millions of times. The first patient with leukemia who treated by CAR-T in 2010. Ever since then, cell therapy starts to enter a new era with the help of the top immunologists and, of course, the local authorities. So nothing is better than seeing a kid who suffered from the leukemia now grow as a young, healthy teenager because of CAR-T. And we have been believed that the cell therapy is and will be one of the best cure for all of the kinds of cancer. So until now, there are more than 2,000 cell therapy clinical trials all over the world, and over 500 cell therapy companies with their own clinical pipelines. There are 7 CAR-T therapy which has been commercialized, including the CytoSinct from Legend Biotech, one of our subsidiary companies. However, due to the price of the CAR-T therapy is absolutely beyond the range, almost $500,000 per treatment, the sales performance I can only say is not as good as expected. And the total sales revenue of CAR-T products in 2021 is only $1.7 billion, which is much lower than other hot products like PD-1, BTK, or ADC. Besides, we know as other cell therapy companies start the commercialization of their products, the competition will becoming more and more fierce. To make sure their own products can outperform other CAR-T products, efficacy is one thing. Another thing is definitely how to control the cost. So let's take a look at the listing price of these commercialized CAR-T products. Even though government from different countries figure the way out by providing the patient an installation plan, still most of us, meanwhile, from the figure on the right, it's clear that the material cost of one dose is almost reaching $80,000, not mentioned about the extremely high labor cost. we have to admit that there isn't too much profit for the cell therapy companies, even though they are like the price is very high. To solve this issue, lowering the cost of the CAR T manufacturing is the only way out. So one of the best way to scale up, to convert the CAR T from autologous to allogeneic. So we all know that the advantage of the allogeneic cell therapy is their affordable price and the short delivery time due to the off-the-shelf property. However, the challenges remain. I think thanks to those top scientists, we already know how to overcome the GVHD for the allo by performing the TCR alpha beta depletion or using another type of the cells like NK. However, there's no clear answer for the challenge of larger sample size regarding to the devices or instrument for each step, such as isolation, expansion, or finish and filling. So among the CAR T production, we can see for each step, there are like multiple suppliers. However, for the cell isolation reagent, more specifically, clinical used cell isolation beads, there's only one supplier. We know there are like countless cell therapy companies, but cell isolation bead supplier, just one. Regarding to the GMP manufacturing process, it is not entirely compliant, since there must be a backup supplier. Besides, we all know the disadvantage of the monopoly. This single supplier can set the price at their own will, because you don't have other options. So not mention that the long and uncertain delivery time of those clinical use isolation beads, so average 2 to 3 months delivery time. Meanwhile, the shelf life of the beads are only 3 to 6 months. So the top left figure includes the delivery time of some products we bought from this single supplier, and the bottom figure is the shelf life of one of their products. So may I wonder, like, how many days left after the customer receiving the beads? 1 month, 2 months, or even less than that? So generally, there are pain points of the supply chain for the cell therapy companies, like monopolized by this single competitor, which caused lack of the backup supplier, long and uncertain delivery time, which make the customer have to use it quick and buy it more often. Meanwhile, we have to admit the technical barrier of this key raw material is pretty high. That explains why there was only one player in the room. Now there are two. We changed the second one. Most importantly, their products are not that perfect. Probably you think it is good enough, but I believe that's the reason you haven't seen the better one. So before then, please allow me to introduce the support from GenScript for the cell therapy process. We first focused on the magnetic beads technology, then expanded into cell isolation beads, T-cell activation reagent, cell isolation instrument, and other products. Our goal is to help the cell therapy companies to lower their manufacturing costs and increase their profitable space. So regarding to the cell isolation part, we can provide a one-stop solution from the isolation reagent, columns, tubing set, and automatic instruments. So specifically, let's have a closer look on our cell isolation beads. The left figure is the specs for our cell isolation and activation beads. The size of our beads is around 100 nanometers, and the whole matrix is not only non-toxic and biodegradable. The right table is the side-by-side comparison between our cell isolation beads and the other vendors' beads. We can find out that our bead cell isolation performance is comparable with Miltenyi, but more economic. Meanwhile, our activation beads show a better expansion rate and a higher undifferentiated amount than using Transact from Miltenyi. Safety here means regarding the certain amount of the target cells, the absolute number of particle or even the iron content for using our beads is less than using Miltenyi's products. As for the stem cell, we know that the main issue for them is the beads cannot even be used for the in vitro experiment or the clinical use. So we know one of the challenge of allogeneic cell therapy is how to overcome the GVHD. The scientists have discovered that this can be solved by performing the TCR alpha beta depletion using the beads. So in the current market, only Miltenyi has their GMP grade TCR alpha beta depletion kits. However, the kit they have contains 2 vials of antibiotic beads and 1 vial of biotin-laden TCR alpha beta antibody. Which means you have to incubate the antibody first with the cell sample, then do the centrifuge to wash and remove the free antibody in some extent. Then add the beads to perform the incubation again, like what I picture here in the top lane. So this two-step isolation is very time-consuming, tedious when comparing to the one-step isolation, where I picture for the bottom lane. And this one-stop isolation can be exactly provided by the TCR alpha beta beads from GenScript. Our TCR alpha beta beads can be directly incubated with the cell sample. So you can imagine this would be more time-saving, easier to scale up, also can give you a better cell status after the isolation, because one-step isolation can minimize the cell injury at a great extent. Because every time like you wash or you centrifuge your sample, gonna lower the viability of the cells. So regarding to the application of our TCR alpha beta beads, it can be used both as depletion and enrichment. The figure A is the facts data for the depletion. 10 million PBMCs were started with the depletion using a GenScript TCR alpha beta beads. After the depletion, the purity of the TCR alpha beta negative cells was described in the figure B, which shows we have the comparable results with Miltenyi TCR alpha beta depletion kits. Except for the reagent, we already used our way to perfect the imperfection. There are still some unsolved issues for the allogeneic therapy. From auto to allo, sample size before the isolation will be 100 to 1,000 times bigger. Normally, for the autologous, total cells of the starting sample will be around 10 to 9 level. But for the allo, the total cells before the TCR alpha beta depletion is around 10 to 11 or even 10 to 12. We know the current isolation instrument can only process amount of the cells for auto. So there's no instrument in the current market designed for the allogeneic cell therapy. Even though authority highly suggests do not split the samples into many instruments, this is their only way out. Allogeneic cell therapy companies have to split their sample and use multiple cell isolation instruments nowadays. Not mention the extreme long isolation time, which will cause the cell injury and low viability. Fortunately, the good news, you have GenScript. We are about to launch our second generation cell isolation instrument, CytoSinct 2000, which is developed for the allogeneic market. The multi-channel design can make sure it has a high throughput, which can meet the needs for the allo market. Meanwhile, it has the ability to help to increase isolation efficiency by decreasing isolation time, which can enhance the cell status. And the fully closed and audit tracking system that it can be used for the cell therapy manufacturing process. So except for the products that we recently designed for the allo market, we also have the current products, which can be used for the autologous cell therapy. Here is a representative application data for the CD4 and CD8 isolation, a side-by-side comparison of cell isolation beads, tubing set, and the instrument between ours and the competitors is shown here. We can observe that our cell isolation solution can provide our comparable isolation performance regarding to the purity, recovery rate, and cell viability. Besides, our instrument can provide the customized process based on our customers' need. For example, if you need a higher purity or recovery by using the automatic cell isolation instrument, we can work with you to realize this point by adjusting the software inside. I don't think our competitor can do that. Their software is very strict and rigid. All in all, like our credo is what you need is what we pursue. So except for the isolation reagent, CD4 and CD8 beads, we would also like to introduce one of our blockbuster products, the Enceed T-cell activation reagent. For the expansion reagent, a better way to show it definitely by fold change, because that is more relevant to the downstream application. So here we choose 3 different donors, including 2 PBMC, 1 leukopak. And top 3 figures are the phenotypes of these 3 donors after the isolation, and the bottom pictures are the corresponding expansion force results. Blue line, red line is the activation results using our beads, and the blue line is from using Allogeneic beads. So here we can find out that our beads have an even higher activation efficiency than Allogeneic, which is consistent with the activation signal data. We also would like to show the memorial phenotype. For the memorial phenotype, we all know that T cells matures from T naive to memory stem T cells, TSCM, to central memory T, TCM, to TEM, and effector memory cell. So normally, less mature of these T cells are more suitable for the downstream application. Thus, from the data here, the amount of TSCM and TCM, those young cells, after the activation using our beads is comparable or higher than using Allogeneic beads. So from the last couple of slides, we can observe that our activation beads are better than Allogeneic transact regarding to the cell expansion force and memorial phenotype. So actually, we were asked so many times that is there any disadvantage for expanding so fast? Or are those GenScript-based activated T cells more exhausted than using Allogeneic transact? So left figure show the exhaustion marker, LAC3, TIM3, PD1 will monitor after activation for 14 days. And there's no difference of the expression of those exhaustion marker between using our beads and the transact. Also, the right figure, the proliferation data. If you focus on the expansion rate from day 15 to day 17, it is obvious that the activated T cells using our beads proliferate much faster than using Allogeneic beads. These results indicated that the activated T cells using our beads are more active after the activation than using Allogeneic transacts. All right, here is the overall picture of our CytoSinct GMP grade products. So our building GMP facility can guarantee the quality of our GMP products in a great extent, so which can support the cell therapy company for their R&D and BLA applications. And moreover, like we GenScript are aiming to build a robust magnetic platform to serve different types of purification and become the leading supplier of key reagent and instrument in GCT industry is definitely our goal. And we would love to partner with the healthcare company to cure the diseases and benefit all the humankinds. All right, thank you. Thank you a lot for your time and attention. I know this session is quite short. Lastly, I would like to mention that most of our products have been tested by multiple external customers. So far, all the beta testing feedbacks are very positive. So yeah, please feel free to ask me any question you want. I think I left enough time for you guys. Thank you.
Thank you, Dr. Yu, for the great presentation. So for all the attendees online right now, if you have any questions, please feel free to drop the questions and type it on the chat box. We will receive the questions and Dr. Yu will be able to answer it right now. So we have a few questions from the floor right now, so let's get started. So the first question for Dr. Yu, it looks like your T-cell activation reagent is better than transact from Mutiny. Do you know why?
Sure. Thanks. So this is a good question. So like we were asked like like every single time. It's like, so like the structure of our, let's pull up the structure of our cell isolation beads is the inner side is the magnetic core, and this is the matrix, and this is coupled with the antibody. And since our activation reagent, Enceed T cell activation, has a better activation efficiency than the beads from Allogeneic, because like our antibody, the antibody we used are different, even though we have the same target, and the way we conjugate the antibody on the beads also are different. I think this is a mixing result, mixing like effect, which can cause our beads is better than Allogeneic. Thank you.
Thanks, Dr. Yu. The next question, you say that your size of your isolation beads is 10 nm, but on Mutany's website, it says that their beads are only 50 nm. Does it mean that your beads are bigger than compared to Mutany's?
Yeah. So yeah. I mean, so like the size of the beads is a distribution. It's not just like a single value. And they're like different way to characterization method to quantify the size of the particle. So we actually did a lot of work. We tried. So when we use the same method to measure the size of the beads from GenScript and Allogeneic, actually they are the same size. Even though I know you guys saw the website, it say Allogeneic beads are 50 nanometers. Actually, when we use inner more recognized characterization method, it is 100 nanometers. The size of our beads is definitely the same with Allogeneic beads. So there's no, there shouldn't be any concerns about this thing. Thank you.
Thanks, Dr. Yu. The next question, can you share some data regarding to the isolation or enrichment of immune cells other than antibody and T cells? For example, NK, gamma, or delta T cells?
Sure. I think I have some data about the NK. So this is, for the NK isolation, the most commonly used beads is CD56. And this is our beads side-by-side comparison between our CD56 beads and Allogeneic's CD56 beads. You see from the data here, we have the very comparable, after isolation, we have the comparable purity and the recur rates. And also we have the data for some other customer actually use the CD3 beads to do the depletion first, then to use the CD56 to perform the enrichment to get rid of those NKT cells. And we also tried to compare those combinations between GenScript and our competitor. And also we have very comparable results. Thank you. And for I think you also mentioned other type of cells. I couldn't remember that. But we have their others other products which aim for the let me show. Yeah, we have here. We have their CD34 beads aimed for isolation of stem cell stem cell. But for now, I'm not liberty enough to share the data at this moment. Thank you.
Thanks, Dr. Yu. For the other attendees, if you have any questions, do feel free to type it in the chat box over here. Dr. Yu will just wait for a while for some of the questions to come in.
Sure, yeah.
Hi, Dr. Yu. Here's another following questions from the audience just now. So the question is, if this is the first time that we wanted to try out CytoSinct, what are the consumables that we need to be purchased?
Okay, so there's like two ways to do the isolation. One is manual, another one is automatic isolation using their this instrument. So I'm not sure which like method you're talking about here. I mean, if you want just to do a manual separation, you can just like purchase the GM and GL column, and also compatible with this magnet and the G stand. And if you want to perform automatic isolation, you should like purchase the tubing set as well as the CytoSinct 1000 hour instruments.
Yeah. Thanks,
and
Dr.
I think I saw any problem. And yeah, I can just like read from the chat box. So here I got a question saying, Transact is washable. Is your beads also washable? Yes, the size of our beads and the structure of our beads is similar with Transact, the activation beads, TSL activation beads from Allogeneic. And washable, I guess you mean the remaining beads, amount of the remaining beads after the isolation. So we did some study on it. So I can just like pull up the data. Just give me 1 minute. So yeah, you can see my screen here. So we compared the remaining amount of beads after the activation, and we put like side by side. Okay, here I think. Yeah, here is some more relevant data. Like we put side-by-side comparison between our beads and also like our beads and after activation for a certain amount of time. So you can see like after 5 days, it looks like there's no remaining amount of the beads after the activation. So main reason is like those beads, the size of beads is very small, it's 100 nanometers. So some part of beads will be washed, like peeled off during the cell culture, and some other part of beads maybe internalized by the cells itself. And you can see like the figure here, we use the MFI of the anti-IgG to quantify the amount of the beads. Because negative control is the empty control, and M here is the is our positive control, and the day 2 is like the day 2 after the activation. You can see longer the time, the amount of the beads becoming less and less, and finally after day 5 or day 5 is the amount is totally like below the detection level, and is negligible. And we compared like this data, like our beads and Allogeneic, and we have the same level. Thank you, but that is a very good question.
All right, thanks, Dr. Yu. Yeah, another question. Yeah, are all the isolation beads for PBMC use? Can we use it for other starting materials, for example, cord blood? If so, do we need to do our own optimization?
Except for PBMC, also you can start from the leukapheresis. And for the cord blood, I'm not sure like in our manual, we just say like it can be used for the PBMC or leukapheresis, but for the cord blood, I don't suggest you starting from cord blood. And for the optimization, you can use the FICO to get your PBMC sample. I think this is a safe way to perform the cell isolation, since the sample, normally sample is rather very expensive. Thank you.
All right, thanks, Dr. Yu.
No problem.
So if there's no any other questions from the floor, we will end the Q&A session over here. So let's say if you have any other questions after this webinar for Dr. Yu, feel free to drop us an email at ap.marketing@genscript.com. We will email back the answers back to you. So once again, thank you, Dr. Yu, for the presentation, and to all of you who joined us today. And I hope you find this session today beneficial to your work. So thank you everyone to join us again and have a nice day. Bye-bye.
Thank you. Bye.